Hi everyone, my name is Yoelys Cruz Leal. I am a postdoctoral fellow from Canadian Blog Service. The title of my poster is Erythrocyte Loss Define Antibody-Mediated Immunosuppression, a study with polyclonal and monoclonal antibodies. Hemolytic disease of the fetus and newborn, FDFN, is a disease provoked by antigenic incompatibility between mothers and fetus. Polyclonal anti-D has been successfully administered to use to prevent FDFN, and this mechanism has been referred as antibody-mediated immunosuppression, AMAS effect. There are several concerns about the future of this therapy, and for that reason, their replacement by a recombinant therapy is desirable. However, the exact mechanism how anti-D work is still understood. The major theory behind AMAS effect is based upon the clearance of the RBC. Recently has been proposed antigen loss as also antigen modulation as a possible mechanism of AMAS effect. The goal of this work was to establish the relevance of the antigen loss as a mechanism to predict AMAS effect. In order to reach this goal, we evaluate the ability of a panel of antibodies to induce AMAS effect as also hot RBC clearance and antigen loss. We use it as a source of erythrocyte, the erythrocyte from the hot antigen mice. This is a transgenic mice that express on their surface a recombinant antigen comprised by het egg lysozymes in tandem sequences with ovalbumin peptide and the human transmembrane protein. How we did that? We inject hot RBC, previously labeled with a fluorescent marker, in the wild-type mice, and 24 hours later, we inject the antibodies, and we bleed these mice at 2, 24, 48, and 72 hours after antibody injection. And we detect the hot RBC in circulation, and also we detect the antigen on the hot RBC recovery, and in the day 7 after the transfusion of the hot RBC, we detect the anti-HEL antibodies by ELISA. As you can see in this graph, we show the correlation between red blood cell clearance and the HEL-specific response induced by aged mice where were injected the hot RBC and each of these antibodies, a panel of antibodies specific against different portions of the antigen. And as you can see, we detected a very poor correlation between red blood cell clearance and a health-specific IgM response. In contrast, when we evaluate the correlation between erythrocyte antigen loss induced by each of these antibodies and the health-specific IgM response induced in the mice that these antibodies were injected, we detect a very good correlation between both factors, because hot-specific antibody induced aims independently of their ability to induce red blood cell clearance, but they cause remarkable erythrocytes antigen loss. In order to explain a little with more details these results, we pick two antibodies, EOVA-specific and polyclonal antibodies, to explain this, how we obtain this data. As you can see in this graph, that we represent the health-specific response induced in the mice were injected hot RBC alone. They induced a high antibody response, IgM and IgG. In contrast, the mice that received the anti-OVA specific polyclonal antibody together with the erythrocytes, the mice didn't induce a potent response. And the OVA-specific antibody also suppressed the health-specific response, IgG. When we evaluate the hot RBC in circulation, as you can see here, the OVA-specific antibody did not induce the hot RBC clearance. The mice show similar level of hot RBC that the mice that receive only of the erythrocytes and PBS. In contrast, the OVA-specific antibodies induced a significantly loss of the Hel antigen and also the O antigen on the surface of the hot RBC recovery. We also did similar experiment with Davie-specific and monoclonal antibodies. And as you can see here, the Davie-specific and monoclonal antibody, wild type, and their desglycoside variant of this antibody also suppress the Hel-specific response, IgM and IgG. And when we evaluate the hot RBC clearance induced by these antibodies, we observed that the wild-type antibody induces rapid clearance of the hot RBC in circulation. In contrast, the desglycosylate variant of this antibody lost the complete ability to clearance the hot RBC in circulation. In contrast, both antibodies induce a significantly the loss of the Hel epitope, and also both antibodies induce a significantly loss of the Davie antigen on the surface. In contrast, that the mice that receive PBS show the same level of both antigen on the surface of the hot RBC. All these results allowed to conclude that red blood cell clearance is not indispensable requirement for AMA's effect, and also antigen loss is a significant predictor of AMA's activity in hot mouse model, and is not complete antigen loss does not seems to be required to obtaining a successful AMA's induction. In order to finish this presentation, I would like to say thank you for all the people in the lab, and also for my supervisor and for the organizing committee for giving me the opportunity to present these results today.